People can turn on their.
Okay, yeah. So we are also going to record the lecture. And so I think we can just start now. And I'm very happy to introduce the speaker today. And I think most of you already are familiar with Karla. So she's a professor of molecular biophysics and biochemistry and also cell biology. And apart from this, she is also the director for the Yale Center for Science and Medicine. And her biography tells that she got into RNA research during her postdoc days. And then since then, her own lab has been interested in various aspects of transcription, splicing, and RNA biogenesis. And it's great that her lab uses a variety of model organisms from yeasts to mammalian cells as well. And I think it's really interesting for our climate group that we could get to talk to her. And we have also discussed her paper. I think we discussed her papers in February, and then we decided that it would be nice to interact with her and talk not only as a researcher, but also as an educator for climate change and how we can integrate education or just principles and philosophies related to climate change into the STEM courses, which actually do not talk about climate change directly, because like she also writes in one of her articles that basic research is usually mostly about treating diseases like cancer and neurodegeneration, but there are also subtle ways where we can include theories on climate change and make students more aware about this. So I think it's really interesting that we have her with us today. And I think, yeah, now I just stop talking and stop sharing the screen as well. And yes, Karla, please.
Yes, hi everyone, and I'm so pleased to join you today. Thank you, Pallavi, for the invitation. I really wish I could be with you in person. Of course, that costs a lot of carbon dioxide to get there. I'm going to Copenhagen today for a conference, so I was actually fantasizing about getting to Vienna, but it didn't work out. Sorry. So I'm going to just make a couple more comments about myself, but maybe after I do that, I wouldn't mind learning something about who you are in the audience. I think we're a small enough group, you can turn on your video screen, at least for the beginning, before I start, giving you a barrage of information about my class. It's really nice. There's Dave Drexel. Oh, awesome. Hi. I've been to Vienna quite a few times. I got a former student, Naos, there. So it's really wonderful to see everyone. And so one thing I'll tell you about my career is that I did most of my career at the Max Planck Institute for Cell Biology and Genetics in Dresden. Well, 13 years of it. I don't know how long my career is going to be, so maybe it will be most, but I didn't have the opportunity to teach. And one of the things that I really loved about my own life was participating in American universities as a student, as a graduate student, and as a postdoc. And so I basically returned to the US. I accepted a job at Yale, wanting to engage with undergraduates and learn how to teach, because, actually teaching from A to Z on a topic is a skill that you need to learn from someone who already knows how to do it. At Yale, there are a lot of teaching resources. So I've learned a lot about active learning, how to engage the students in the material. And so I feel like I've got, become a bit of a card-carrying teacher. And realizing that I had some teaching skills and I had an audience, I finally realized, what I could do with that. And that was to develop a class on climate change. And so that's what I'm going to be telling you about today. But I'm curious to learn who you are. How many of you are biochemists? I'm also a neuroscientist. Is somebody not a biochemist? Maybe somebody's got another specialty, a neuroscientist or an engineer. Are there any engineers here? Environmental engineers, ecologists? You're an ecologist. Woohoo! What do you work on?
Hi, I'm mainly working on, let's say, environmental microbiology, a lot of climate change research. Yeah.
Awesome. One of the things in developing this class that I'm going to tell you about, I really gotten so interested in microbes. They really rock the world and are so important. Also, my students are really taken by plants. So let's see. Has anybody else tried to develop a class around climate change? Has anyone? Okay, great. What was the class?
I decided to not preach to the converted, but just integrate it into a regular course. Hello, I reached out to you after your wonderful article, and I have a bachelor class for molecular genetic genetics, and I decided to teach about climate change and reproduction. And I think it was important to put it in.
That's a great topic. There's just a big article in the New York Times about how girls are going to puberty 3 years earlier than they should because of plastic things. Well, we can talk later about what you have in your class, but Peter, okay, so Peter, thank you for emailing me before. And what about Jerome? Do you teach in a different class?
Yeah, I developed a course on sustainable lab practices, more or less, which I'm teaching at the Max Perutz. Yeah.
Nice. So I would love to do that at Yale, reduce the garbage that we make in our labs. Is that part of what you're on about, or?
It's more giving the background, right? What is the footprint of lab practices? How can they calculate the CO2 footprint? And then also looking a little bit more in a broader scale at the academic sector, mobility, and the usual topics. Yeah.
Okay, well, we don't have that class here, so I would love to have that. So one of the things I go around and talk about this a bit, if I give a seminar in RNA biology, which is what I work on, then I also ask to speak to people about my class. And at least in the US, what I've noticed is the only people who come are graduate students and postdocs, and the faculty don't come. I'm basically giving them a roadmap to make a class like this if they want. It's an upper level elective. So they've already had 2 semesters of biochemistry. In the first semester, they've had, a lot of sort of chemistry, the chemistry side of biochemistry, metabolism, amino acids, nucleic acids, structures, and so on. And then the second half, it's basically central dogma, DNA, RNA, and protein from prokaryotes, a lot of classical experiments, ending up with immunology is like in the last lecture. So it's quite the tour. So this is an upper level elective, and anybody could add to their department's curriculum in biochemistry. And I'm happy to share materials. So if anybody wants materials. So, okay, well, listen, I'm going to start going through some slides, but please feel free to ask. I've arranged myself so that I can see everyone. And if you want to ask a question during the talk, please ask a question. I'm just going to be touching down and giving some examples about the class, and following on Peter's comment about preaching to the converted. Okay, wait, I've got to share my screen. Somehow there we go. Okay, so hopefully you can see my PowerPoint there, and I'm going to save myself by not going into presentation mode. Yes. Okay, I was excited about this title. I'm going to stick with this for a while. A new dawn for biochemistry. So, as Pallavi mentioned, so I decided in the 90s, all my hobbies are outdoors. I love wildlife, plants. I'm a gardener, all these things. And when I read an article by Thomas Keneally in Australia when I was camping in the outback in Australia in 2006. And it really, of course, I'd heard about global warming and climate change, but this one really changed my life because it made me want to do something. And I felt like I'd gone into a specialty where there wasn't anything I could contribute. And that lasted for quite a long time until I recognized that I'm actually a teacher. So I do think that it's a new dawn for biochemistry. And so here's a really important point. We need the next generation of biochemists to develop things like enzymes that can make compounds that are renewable. In other words, something that could make a plastic. I'm going to give an example of that in a minute. And then we need enzymes that can then restore that plastic back into monomers so that we can take an organic compound that polymerizes and make a plastic window, a polyacrylate window. I'm going to show you a company that's doing that. And then when the window is broken because somebody threw a baseball through it, you take the shards of glass back to the factory. They use an enzyme to break it back down into monomers, and you can remake a window. So you're burying carbon dioxide in a window, and then you're going to recycle it back into windows. And so we can bury a lot of carbon dioxide in buildings if we harness the power of biochemistry. So what motivates me is that biochemistry is awesome for addressing a lot of questions about climate change, but the next generation of biochemists are 17 years old. And at least in the US, I don't know if what I'm saying is exactly relating to the European educational system, but in the US, nobody tells you in high school how you can contribute to meeting the challenges of climate change. Kids, and I heard my own son saying this to his friend Max Hyman, I want to save the world, but from climate change, but you have to be an environmental engineer or a policymaker in order to do that. And I'm listening to that, and that was like the watershed moment when I thought, that's crazy. Everybody has something to contribute, and certainly biochemists have something to contribute. And if those 17-year-olds don't go into biochemistry, which they might be interested in, then we're going to lose the next generation of talented biochemists. So we have to make it clear to young people how our discipline that we love and we know is powerful, we can make clear to them what it has to offer. And a lot of students in my class are pre-med, and they end up going to graduate school after my class because they're like, holy cow, I could make an impact here. And it's not that I'm against going into medicine. It's just that people need to follow their passions. People are passionate about climate change. People are afraid about climate change. And to some extent, a graphical word is coming to some extent, paralyzed is what I want to say, paralyzed in making career decisions about following their interests, because there's this tug on the one hand of want to do climate change, but on the other hand, I'm really interested in biochemistry. Okay. So that's how I think that a new dawn for biochemistry is to just get people interested in biochemistry, to realize how important it is and get them to pursue a degree in biochemistry. Okay, I'm going to shut up. So all of these are images, sort of apocalyptic images that you all know. There's a question from Nicola.
Yeah, I just wanted to say we are not in the presentation mode, so we see your slides.
What?
We see your slides not in the presentation mode.
I know, but I don't want that, because then I can't control my world. I can't see you very well if that, is this okay? You see my slide, it says life during wartime. I'm quoting the talking heads. Is that okay for you guys? You can't see it properly? Okay, good. So these are the apocalyptic, visions for what motivates us. And then we have to dig into to how we're going to address things. So this is actually a book that was really important to me, and I recommend it to anybody. You can recommend it to people. It's All We Can Save, edited by Ayanna Elizabeth Johnson, who's a marine biologist. And her attitude is everyone has something to contribute, and you just have to ask, what are you really good at? And apply that to find a way to make your contribution. So my contribution is in teaching. And whatever donations I make. Okay, so so the question that I address in my class is, what is the biochemist's role? And now what I'm going to do is tell you how I engage the students at the beginning of the class. So this is a slide from my first lecture, and my point is that everything in biochemistry depends on temperature, the concentration of salts, and the pH. And all of those things, particularly for marine or aquatic organisms, are changing drastically because water is absorbing more of the temperature than air. And so we really desperately need to pay attention to those things. And of course, there's heat stress for land and aquatic animals, among other things. So I realize that you guys are all the converted. In the one thing that's really interesting in the US, and you could reflect on your own system, is that the CDC, the Center for Disease Control, has embraced the One Health concept. And that is that you can't cure a sick human if the planet is sick. We need to have wildlife. We need to have agriculture. We need to pay attention to the interaction between humans and animals. We have zoonotic diseases that are definitely going to be moving around in response to climate change. And so CDC recognizes this, and it's on the front page. Like if you go to the CDC, the problem that I have and where I think the root cause is, and this is in my first lecture, because I'm talking to these students who think they're going to go into biochemistry either to become a doctor or to cure human disease. Where do we get this bias from? In the US, we get the bias from the fact that we have to get money from the NIH. It's the National Institutes of Human Health, not planetary health, human health. And it's just a list of organs. Look at this. Well, we start out with cancer. That's great. It's great to fight cancer. Eye, heart, lung, blood, the genome, aging, alcohol abuse, allergy. These are none of these things apply to the environment. The only one that applies to the environment is this one, the NIEHS, which is the most poorly supported institute. It's pathetic, the amount of money it gets compared to all the other ones. And it, look at when it was, Rachel Carson, man, she did a lot of things. So the NIH does not, in order to get money for my research, I have to say that pre-mRNA splicing is important for cancer, which it is, but I don't have to say that pre-mRNA splicing is important for adapting to temperature, which it also is, because the NIH doesn't care if pre-mRNA splicing helps a mouse or a human or a Drosophila or a fish respond to heat shock. It's not interesting. So I have to say over and over and over again, it's like an affirmation that my work is relevant to human health, neurological disease, cancer, et cetera, et cetera, et cetera. I have 4 different R01s from 4 different of these institutes, and I'm capable of saying these sentences to get money. But the problem is that it impacts me. It makes me think that my work is only relevant to these organs. And now I've realized that studying stress is also relevant to algae. So we're studying algae in the lab, and we're just not going to tell the NIH. We're going to try to get money from the NSF. Okay, enough about me. But I think that's where the problem comes from. So the goals in my class are to engage the students of today and tomorrow by finding and exploring biochemistry topics central to climate change and its relationship to the living world. Recruit and train the next generation of biochemists. They can opt to work in translational planet medicine and save the world. I was going to say something else, but I forgot. Okay. Did you have a question, Peter? Okay. You're just scratching yourself. Okay. So this is my first slide. This is my favorite place on the planet. It's the Upper Peninsula of Michigan, and it inspires me a lot. These are a bunch of students who've worked on this class with me. So I recruited. So, I know what I wanted to say. Engage the students to explore biochemistry topics central to climate change. There's no textbook for this, right? Like Peter knows about this. If you're going to try to teach a topic, you have to make it yourself. You have to find papers, like a crazy paper about how different degrees of temperature change alternative splicing. You have to find that paper and then use those papers to construct a climate relevance for that biochemistry. And then the student can write a mini review or a final paper that elaborates those ideas. Like, hey, I wonder what happens in algae when they experience heat shock in the context of coral bleaching. You could ask, what happens to gene expression? But nobody, so you won't find that paper. You won't find the answer, but you can find enough information about biochemistry to ask those questions in your paper or in your research proposal, which is what my students end up doing. So the students in my class are doing exactly what I'm doing to make the class. They're looking for topic areas and they're following their interests. I'll give you an example right away. This seminar could go way over time. So I'm just going to keep talking and then we'll cut it off. Okay, here was a cool research paper that someone wrote. She found some literature on plants that transport ammonia out into the soil and end up burying calcium carbonate. I told you, I get really excited and then I can't say the words properly. Sorry. So they transport calcium carbonate out into the soil. These are plants that actually kind of fix oxaloacetate. They emit oxalate into the soil where it forms little rocks. So they're actually sequestering carbon in the soil, these particular plants. So I never heard of that. So she found out about this and she wrote a whole term paper trying to isolate. She proposed to isolate the transporter that transports calcium carbonate out of the root into the soil using oocyte injection. She was going to make a cDNA library and inject it into oocytes and find the transporter, which is a really brilliant idea. It's a great experiment. And so the students are basically doing what I had to do to make the class. So these are students who really helped me a lot, the first TA, the second TA, and this undergrad who I hired during the pandemic to do the research. So in lecture 1, I give an example. I have them watch Australian Broadcasting Company makes nature shows that are really very scientific. They explain a lot of experiments. So this is, pop science, but it lets you ask the question, what is coral bleaching? So I have them watch the video and then everyone says, well, obviously coral bleaching is when the temperature goes up in the ocean and then it kills the coral tissue. But as biochemists, we want to know what is the mechanism behind that. There has to be a molecular mechanism that can explain why the coral tissue dies. And turns out that it's because the symbiotic algae inside the coral tissue, this is a symbiosis. Yeah, most of you know this, but there's a tissue inside the calcium structure that you normally see as the coral reef. And within the tissue, you have symbiotic algae that give the coral the color. And they're doing photosynthesis and making the food for the coral tissue. And what happens is because photosynthetic organisms are more sensitive to UV and heat, because they have all this electron transport and massive, water splitting and oxygen flying around, they experience ROS, reactive oxygen species, when the temperature goes up. So this is their stress response to the temperature. So I teach them about ROS in the class. What they end up doing is exuding peroxide, and the coral tissue squeezes the algae out of the tissue because it's like having bleach poured on them. They squeeze the algae out and now they don't have a source of food. So that's why they turn white. And if they don't get repopulated by algae, then they will starve to death. And that's a simple explanation for coral bleaching. So then you could ask all kinds of biochemical questions about the algal symbionts and how you could, potentially have them adapt so that they don't undergo ROS, or, I'm sure the people on the screen their imaginations are going wild. How could we work with algae to ameliorate this situation in the context of coral bleaching? Okay, so this is my first lecture. I go through the coral bleaching and show that as an example. I talk about oxidative stress, antioxidants, and the temperature sensitivity of photosensors. And then you can see the other kind of topics. And so I'm going to touch down on some of these. Which ones are my favorite? Honestly, they're all my favorite. My class usually starts with, this is my favorite lecture. So I talk about extremophiles. These are organisms that live at extreme temperature, pH, or salt. So I usually start the class with a slideshow of amazing organisms that live on snow. Like there's a red algae that lives on snow. It actually causes heat absorbance for the planet. So it's not good that red algae live on the top of snow. I end up talking about molecular chaperones and how they're tuned to temperature. So I'm going to show you some slides about this. Maybe one thing while I'm showing you this slide that I can mention is that at Yale, the introductory biochemistry class does not cover photosynthesis. It does not cover nitrogen fixation by nitrogenase. It does not cover aromatic amino acid biosynthesis. So these are the reasons we have life on the planet, and they are not covered in a biochemistry class at Yale, which I find astonishing, but now they are covered by my class. Is anyone else appalled by this fact? I'm looking at David. I'm appalled by this fact. And the reason they are not taught is because they're not on the MCAT, the standardized test for getting into medical school. I have no words. Okay. So usually what I do, I have a lecture. So I have like 12 to 14 lectures in the semester. I have the lecture, which is the climate motivation for the thing. I'm going to give you examples. And then I go into the biochemistry. So where I'm showing structures, catalytic mechanisms, iron sulfur clusters. It's very biochemical in the middle of the lecture, but I decorate it. I frame it with all of these reasons why you should be interested in nitrogen fixation. You know, on the outside. And, you know, it's like a chocolate with caramel on the inside. So the biochemistry is the caramel and the climate is the chocolate on the outside. So, so I give them the information that they need to talk about a paper, a single paper in the discussion section. So they do a write-up, and the students have told me that that they like my class the best for learning how to read a scientific paper, because I only assign one paper. I ask them the exact same questions every time. So they have to do a write-up before the discussion section. And the first question is, what is the hypothesis that motivates the study? The students have the hardest time with that question. They go down some rabbit hole, you know, about like, oh, what's addressed in figure 5. But like, why did they do this study in the first place? They wanted to know, you know, why algae are stressed during when the temperature goes up, or, you know, something like that. And the students have a hard time identifying those things because they're trained to get into the weeds. They're given all these detailed information about structure and function from the top end, right? We already know the answers. Then we could go back and look at how somebody figured that out, like, you know, Watson and Crick or something like that. But they don't get that front end piece of a paper where the people say, okay, we know this, but here's something we really don't know. Like, what is that? And so they learn how to do that and then recognize how the methods, the biochemical methods, when you do all those things, allow them to address that question, you know, from the point of view of not knowing the answer. And so I let them just do one paper. I give them a support paper on mass spectrometry or whatever the heck we're talking about. And then they write this 1-page write-up. And by the end, the write-ups are great. You know, I ask, what's the strongest, what's a strength or a weakness about this paper? What's the key experiment that underlies their conclusions? You know, so in the beginning, they're actually remarkably, you know, it was really struggling. These are great students, but they really learn this by just concentrating on this one paper. So then we do, and I'll give you an example of the types of papers that we read. I can give the reading list to anyone they want it. 4-page mini review they do in the middle, and then of the class. So this is something where, let's say, the woman who realized this carbon fixing plant, okay? So she could write a mini review about the literature on that. And then when we come back from the spring break, they give talks, flash talks on like a 5-minute flash talk on their mini review. And then everyone gets to hear what they learned and ask questions. And so they often, some students aren't so great at writing, but they're brilliant at public speaking. So it also gives people a chance to show those skills. In the final paper, they do like NIH 12-page proposal for experiments where they have specific aims and so on. And we workshop how to write the abstract and also, you know, the specific aims and so on. So I kind of stage the exercises before they have to submit their final paper. Does anybody have any questions about the assignments? I don't have any tests.
I have a question.
Yeah, Peter.
How many students do you have in your class, and what's the level of the students when you actually get to have them?
Right. So I tried to explain there, this is an upper level class. So in the US, intro class is 100. They've already taken 300, 301, which is our core course in biochemistry. The first semester is very chemistry oriented and structure, amino acids, nucleic acids, protein structure, and so on. And then second half is central dogma type stuff. So, and that's a really hard course. It's notorious. This and the students, they're very good. They're really very good. Okay. And then how many students? So great question. I've given this class now 3 times. It's turned into the most popular class in our major. So the first couple of years we had 15 students, and we wrote that publication, 5 questions on how biochemistry can meet the challenges of climate change. So that was a class assignment. So we just broke it down in the class. We said, okay, we're going to have this many paragraphs, we're going to have this many sections, and then people collaborated on writing the sections, and we published a paper. So that was great. So this year I had 40 students, and I didn't know what to do. I thought about capping the class because a lot of things had to be changed for 40 students, but I thought I don't want to exclude anyone who wants to hear about this. So I had to get a bunch more TAs because grading all these writing assignments is very time consuming. I also didn't tell you about the field trips. So we go on 3 field trips. We go to a botanical garden at Yale so that they can see the succulent plants that we discuss. These are plants that are, drought resistant. They have special mechanisms for sequestering water and so on. They're CAM C4 plants. So we go to the botanical garden, and they get so excited. It's the first time they've seen a plant. I mean, a lot of these students, unbelievable. They're like, oh my God, plants are so cool. It's like, where have you been? So then I took them to a wastewater treatment plant, 160 million gallons a day at the New Haven sewage treatment plant. Huge microbiology experiment. So where's that microbiologist who we were talking to? So that is amazing. It's, the centrifugation is 1G. Nobody understands when I'm saying that. The shit has to fall to the bottom, And so you get it to do that by flocculating with the right kind of bacteria that glue themselves onto the poop. And then they fall down at the 1G, to the bottom of the thing. Anyway, it's just amazing. It's just amazing. And in today's world, people are harvesting, basically the remnants that you get out of the wastewater treatment plant to make other compounds for building materials, for example, believe it or not. We could talk more if you want. And then the third one is I take them to the forest. We go birding, and because the warblers come through New Haven in April. This is really emotional for me because I get to know the students really well, and they're very moved. I get 40 pairs of binoculars, and I take them out in the forest, and they look at the birds, and they get really excited. And we also, I'm really good at finding tardigrades now. So we hunt tardigrades, and we go back to my house and we look at tardigrades, and we eat pizza. And so we're gonna be doing this over the summer. A lot of the students stay for the summer and do research. So they get really excited by being exposed to nature and by seeing organisms like tardigrades that can survive desiccation. And we talk about the proteins that enable that. Okay, I better get off this slide. Okay, so so this is like I'm just giving some examples. Like this is the beginning of my lecture on the effects of temperature on gene expression, where turtle sex determination is really a classic. So they can take everything they know about chromatin. It's that the temperature alters the signaling that changes chromatin histone tail modifications. And that determines whether you're a female or a male. And with temperatures going up, the danger is that turtles will go extinct because males are undergo sex determination, will become male if it's cooler, in a cooler temperature versus the females. So that's a topic that's really accessible to them. I also talk about changes in transcription and RNA processing as a result of temperature change. So we can really dig into some follow-up biochemistry from their last class, and where they can really apply what they've already learned. So this is an amazing paper from actually a lab in Berlin, where Florian Heid showed that actually there's an exquisitely sensitive catalytic center in a kinase that phosphorylates SR proteins, which I did my postdoc on. This is all very kind of ironic. That responds to, even in us, circadian temperature differences, where we have different alternative splicing programs in our bodies as a result of 1.5 degrees difference in temperature. And that's because this catalytic center, like, literally like, the whole protein remains structured. Obviously, look at those temperatures. But the catalytic center is very sensitive to the temperature, and the kinase is shut off by this small change in temperature. It's super cool. Anyway, so then I talk about extremophiles. So here's an example. I talk about the algae that grow on the bottom of Arctic ice and feed the krill, which are at the bottom of the food chain. We wouldn't have whales if we didn't have ice. So I don't know what's going to happen there. What are the krill going to eat? We can worry about that. They're amazed by antifreeze protein, which is an intrinsically disordered protein that becomes ordered when there's ice around, because it recognizes the pattern of the polarization in the ice crystal. It's so cool. When they find out that there are water channels and that there's an ice binding protein, they're just gobsmacked. They just think this is the coolest thing. So we talk about that. We talk about chaperones. They get the basics of protein refolding and folding in that basic biochemistry class I told you about. But we even look at these, how the chaperones themselves are tuned to temperature. And so then you can see that thermophiles tune their chaperones to a higher temperature in order for the whole organism to be tuned to a higher temperature. And there's all kinds of things you can imagine once you know that, right? So that whole lecture has a big core on the structure and the function of chaperone, the chaperonin complex. I mean, it's I'm just grazing here. Suspended animation is this very cool phenomenon where animals like tardigrades can go into quiescence. And the tardigrade is amazing because it's a full-on organism with a head and a tail and those cute little legs. And when you desiccate it, it just desiccates down into a so-called ton, and it can live for 100 years. You can kill them, don't worry. But they can desiccate into a ton that lives for a very long time. And then when you add water back, they rehydrate in a matter of an hour. That's what we're doing when we're eating pizza. They're rehydrating, and then we can go look at them wandering around in the water. They're so cute. And so that's so cool. So that's suspended animation. They basically have lost their metabolism. Their metabolism is down to almost nothing, but they are not dead, right? And so this is a whole lecture having to do with largely heat and desiccation survival that seeds have to do all the time. Tardigrades do it, nematodes do it in the soil. And we talk about the glass transition that these undergo when intrinsically disordered proteins that get expressed in a sequence, right? So you can't immediately be desiccated. They have to recognize, they have humidity receptors. Like, how amazing is that? There's a receptor that tells you you're being desiccated. And then you start expressing these intrinsically disordered proteins that will enable the glass transition that will protect membranes and proteins and all the constituents of this cell, so that when you add water, you will be alive. It's amazing. Okay. So they love that. We talk about forests in one of the lectures, and that's really exciting because then I get to hammer on Monsanto basically and point out how evil they are. because Monsanto develops Roundup, you know, and it's every year there's a new Roundup. And how does Roundup work? Does anybody know? Okay, so you're all biochemists. You want to put something on leaves that causes that plant to die and nobody else to die. And you don't want you don't want animals to die because then humans would die and that would mean you're out of business. So we can't have humans dying. It could be bad for them. We don't know. We actually still don't know. Okay. So what is something in plants that plants have that humans don't have that we could attack in order to kill those plants?
The cell wall, maybe.
Hey, what?
In the cell wall, something.
Actually, that's a really good point. I'll come to that. No, they have
Well,
aromatic amino acids. They have aromatic amino acids. Do you know how many enzymes it takes to make aromatic amino acids? It takes 25 amino acids. So we have the shikimate pathway. I'm not going to drag you through this, but there are many. See these numbered? There's sort of several different cycles of synthesis that you have to do here until you make chorismate. And then this blah, blah, blah goes through all of these different possible pathways in order to produce tyrosine, phenylalanine, and tryptophan. Okay, so plants will die if they don't make the aromatic amino acids. We won't die because we'll go down to the grocery store and get some aromatic amino acids. So this is their playground. Monsanto just targets all of these enzymes that make wonderful aromatic amino acids for us and kills the plants. And so we can talk about, you know, glyphosate and how it sits in the catalytic center and it looks like one of the substrates. So isn't that terrific? And so there's this just big, you know, we talk about the economics of the whole thing and how the farmer is caught in this trap where they have to buy Roundup and because they have to buy the wheat that's a GMO that's allergic to this particular kind of Roundup that they have this year. Terrible thing. Okay, so we talk about soil health. We do nitrogenase. So this is really a reach for me because my inorganic chemistry is really tough. But so I've been improving my inorganic chemistry, which I haven't studied since the last century. But it's a lot of fun. Nitrogenase is a really cool enzyme, and like I said, we talk about plants that are succulents. Here's our field trip to the botanical gardens. That was 2021, everyone was wearing a mask, where the students really become very fascinated by the plants. And in fact, it's very cool because you can slice across the leaves. If any of you teach about CAM or C4 photosynthesis, which is a mechanism for leaves to keep their stomata closed. When they open their stomata, they lose water in exchange for carbon dioxide. So they will, if you leave your stomata open all day long and you live in the desert, you're going to die because you're going to lose all your water. So they've evolved mechanisms for, they open their stomata at night to capture the carbon dioxide, and then they sequester it either in another cell or another compartment in the cell. And the way they do that is to make a 2-carbon sugar malate. And that 2-carbon sugar can't go anywhere, right? Whereas carbon dioxide can diffuse back out. So they capture it and then use that to enable them to fix carbon, you know, around the clock by doing their own version of carbon capture. So it's easy to see these specializations in the leaves. You just slice through the leaf with a razor blade, honestly, and a 10x microscope bright field, you can see these specializations. So it's easy to make a demonstration for the students, and they'll recognize them right away because you'll give them the background for that. Let's see, I wanted to tell you, okay. So here I'm, this is one of the ads that I made. So when I created the class, I advertised, I had people send out these little ads. You'll see them all right now. So this one is what's in smoke from forest fires that causes seedling roots to grow? So this is the control, and this is adding an extract of smoke, and you can see that the roots are longer. So the obvious hypothesis is all of the forest fires that may be caused by climate change, at least if the smoke that's given off, the rain comes and brings the compounds that might be in smoke back down to the earth, then there may be things in it that assist in regrowth of plants in the burnt areas. And it turns out carbachol is the molecule, and it was identified in Baldwin's lab in this paper, actually. And the paper, they love this paper. It's a really great paper. They do, chemical fractionation. You test all the fractions because you have a clear bioassay, which is the root growth. They characterize the molecule. They in order to prove that it really is in soil after a burn, they go and they collect soil from a place in Arizona that just had a forest fire. There's some really kludgy things about this paper. One thing is that they use, so you imagine that they're making an extract from forest fire smoke. In the whole paper, you're imagining that. And this is how I get them. This is the second paper we read, that I get them to read the methods. So I say, how did they extract the smoke? Did you read the methods? And they're all like, oh God, I didn't read that. So then they're all flipping through their pages, looking in the methods, and oh, it's liquid smoke, they say. And I'm like, well, how did they make the liquid smoke? And they just say, well, it's just liquid smoke. And then they give a source, which is a spice shop in Piscataway, New Jersey. Does anybody know what liquid smoke is? It's an ingredient in barbecue sauce. It's like made in like hickory smoke, it's in barbecue. That's what makes the barbecue sauce. It actually is an extract of smoke, but it's a spice, and that's what they use as the input. So the students actually learn the lesson that they have to pay attention to the methods. And that's why it was important that they went to Arizona after a forest fire to show there was carbachol in the soil, high levels of carbachol. Okay. So then I have a zoonotic diseases lecture, and what I'm talking about in that lecture is how the insects know to bite us. So it turns out they're, very adapted to biting humans. Actually, they prefer humans to any other warm-blooded thing. And so what they like is they like our carbon dioxide that comes out of our mouths and lactate. So when you go for a run, you're going to attract more in your sweat, more mosquitoes. And so there are lactate receptors and carbon dioxide receptors that have been characterized beautifully. There's really beautiful neurobiology about how insects sense us, how they what DEET does. So we read a paper on how DEET binds to these receptors and works. So this is also a very cool area. I do a fake meat lecture where I can bring in how do we taste meat? There's an umami receptor so we get to follow the chemosensation of the insects with our own chemosensation and talk about the umami receptor and the fact that what makes meat taste like meat. I almost can't look at this, I'm vegetarian. I'm going to switch to the cow. I like the cow better than the hamburger. We talk about how hemoglobin is what makes meat taste like meat. To us, we don't know how we taste hemoglobin currently, so that's really an interesting topic. Some people call it a GMO because they make soy leg hemoglobin is what they use to make the Impossible Burger. So what does that mean? It's legume hemoglobin. So let's talk about why they use hemoglobin. Why do we use hemoglobin from soy leguminous plants? Anybody know? Okay, here's a clue. It comes from the roots. Okay, here's another clue. So legumes are nitrogen fixers, and they fix nitrogen because they make these rhizomes. They have a symbiosis with bacteria, and the bacteria doing the nitrogen fixation, and the root nodule around that includes the bacteria are an adapted space for the bacteria to fix nitrogen. Nobody's raising their hand. The last clue is nitrogenase is not a very good enzyme because it can also bind oxygen. So what you'd ideally like is you would ideally like an oxygen reduced environment where you prefer your nitrogen would occupy the nitrogenase more often. Soy leg hemoglobin binds oxygen, binds heme, sorry, it binds heme 10 times more tightly than our hemoglobin. So it's making an oxygen reduced environment for the nitrogenase in the root nodules. It doesn't have blood. Plants don't have blood, but they need oxygen binders to suck the oxygen out. A lot of things are done really locally. Like, think about it. Carbon dioxide is in the cytoplasm of the plant cells. I mean, how does it get the carbon dioxide? Like, all these things are dissolving in the fluid. So that's what you're working with. You're just trying to create a local environment. Anyway, no, David seems somewhat interested in what I just said. Okay, anyway, so here's this fun thing. I'm looking at the time. Don't worry, I won't go over. How does eating red algae keep cows from belching methane? So does everybody know this one? Okay, so a Canadian farmer has his cows. He's on the west coast of Canada, and he noticed that seasonally there are times when there's a lot of red algae that comes up on the beach, and the cows end up eating it. And the cows were belching, somehow less. He thought their health was improving. And I don't even know if that's true, really. But the point is, somehow they mentioned this to a biochemist they knew, and the biochemist figured out that they weren't making methane anymore. And so what you need to know is that in the rumen of the cow, 4% of the microbes are archaea. Archaea are the organisms that are methanotropes, and they make methane. Okay, so the archaea, that's 4% of bacteria in a cow's normal rumen, are oozing methane. Methane's much worse than carbon dioxide. And so there's something in these algae that kill or stop the methane production. And it turns out it's bromoform that inhibits this. And so I end up teaching in that lecture is methanogenesis. And the reason why that's very cool to teach is there are a lot of enzymes involved in that pathway. So you can imagine, instead of feeding your cows bromoform or red algae, you could have a chemical inhibitor, let's say, of an enzyme, because actually bromoform isn't very good for you. It's kind of toxic. So the students get to ponder that idea that you could have a chemical inhibitor of an enzyme in the methanogenesis pathway to reduce methane production by, farm animals like cows. Okay. So I noticed that people are getting restless, and I wonder if I should wind up. Does anybody want to say anything? I was going to give this example of the windows that I think is really exciting. Can I give the example of the windows?
5 minutes, yeah.
Yeah. Okay, so I think my most popular lecture is the one that's about architecture and how we can bury carbon in buildings. And the students start out, so it turns out this massive wood building was started in part by an architect in New Haven at Yale. This is a total accident. My daughter's an architect, and she told me about this. And then it turned out the guy is in New Haven. So I go down to his office and I get these massive wood samples and I show it to them. So the idea is we should bury carbon in buildings, because once it's in a building, it won't decay. Planting a tree that's going to fall down and decay is just going to return the carbon dioxide to the atmosphere But if you take wood and put it in buildings, then you will store the carbon. And another way to store the carbon, so you can do like this, you can make all these materials, like you can make a wood masonry versus brick masonry. The brick masonry costs carbon dioxide, and the wood masonry sequesters carbon dioxide. So the other way that we can store carbon dioxide in buildings, I mentioned this before, is with renewables, and this requires enzymes. So biochemists should be doing this. If the man walks up to me on the street and says, how can I be a biochemist and save the world from climate change? I'm going to tell them, understand enzymes. Because here's an example. A friend of mine is the CEO of Arzeta, CSO, sorry. Okay, here's a molecule and an organic compound called tulipolin A. It's from tulips. If you had a lot more tulips that are in the world, you could make this compound a lot. You could just purify it from tulips, but we don't have enough tulips for what we want to do with it. We want to make windows out of it. It polymerizes spontaneously into this clear acrylate that has all of the same properties of glass, except it's not quite as breakable. You can think of all the windows and buildings that we need to house the population by 2050. It will take our entire carbon budget to make those buildings. If we would stop having wars, we would do ourselves a favor. Okay, we could use this polyacrylate to make the windows. And like I said before, you can, when they break, you can take them back to the factory. And if you had the right enzyme, you could degrade the polyacrylate back down to the monomers, and then you could remake a window. So what they're doing in this company is they're using neural networks to figure out which enzymes could potentially make tulipolin A, because that's actually not known in the plant what the enzyme pathway is for making tulipolin. So then what they do is they just make tons, bacterially expressed enzymes, and they do experiments to try to make tulipolin A. And then the second half is isolating an enzyme that'll degrade tulipolin A back down to the monomers. There are protein engineering cases, which is what I teach them in the class. At the end of that class, I journal club a paper that takes an enzyme from an insect or a microbe that degrades plant materials are really hard to degrade, and they're carbon oxygen rich, just like all plastics. So that's where our plastic degrading enzymes are coming from, is from wax worms, microbes in the termite gut that degrade lignins. You need really hardcore hydrolases, cutinases, and so on that can degrade these proteins. And then you can evolve them to be more efficient to be used on industrial scale. So this is a very inspiring class for the students. And they usually write me an email after the class because they find it very inspiring. Okay, so I know it's 10:59 for me, and that means it's 4:59 for you. So you want to go and have a beer or something. But are there any questions that I could help address?
Yeah, I think maybe we can begin the discussion, so that if you have still any relevant slides concerning the discussion, then maybe we can continue another 10 minutes.
I'll just put I just have endless number of slides.
Yeah.
So I'll just put that up. But if people have specific questions or want me to send them my syllabus and stuff like that, I'm happy to do that.
Yeah, but also thank you for this very exuberant talk. And I think we all really enjoyed listening to it. And I think it was also sort of very eye-opening and brain-opening, in the sense that it kind of changes your perspective to look into, old biochemistry textbooks like Stryer or Leninger, and then also just things around you. And I think it will be really useful for a lot of researchers who are also actively teaching. So,
well, I mean, one example is these intrinsically disordered proteins I told you about from the tardigrades that enable them to do this glass transition.
yeah.
You can purify those proteins, lyophilize them together with enzymes, and they preserve the enzyme. So the enzyme is more active when stored like that. So here's how that's climate relevant. All the people who need medications that need to be refrigerated. How about the Moderna vaccine? It needs refrigeration. you can use all these compounds from tardigrades to protect RNAs and proteins that you're trying to deliver to patients in regions of the world that are hot and getting hotter and don't have refrigeration. So, you know, these are kinds of things that the students latch on to immediately. They can immediately see how being a biochemist is giving us power to address real world problems and face climate change.
I think it's also really important as an educator to really tell these things to students who are just starting research, perhaps like, you know, you do for the undergrads, but maybe also for people who are also actively researching, like, let's say, perhaps talk not only about the phase separation of these proteins, but maybe from a climate perspective as well. So I think that would be really interesting.
I'll write back to you and tell you the first time I get a grant for my studies on algae. It's gonna be, might be a while.
Yeah. But yeah. So where do you think the actual bottleneck is then to sort of maybe integrate more of these philosophies or these kind of, you know, concepts?
I still think in the US it's funding. So let's say one of my students wants to go to graduate school and become a climate biochemist. They will have to go to a lab, you know, they're like, so people come up to me and they say, how do I find a lab that does stuff like this? I mean, you can go to David Baker's lab where they do a lot of protein engineering. You know, I would suggest go to the tardigrade guy's lab. You know, you have to take, you know, maybe some of these examples where it really looks like the lab is concentrating on this question, and then maybe try to go to those labs, or just recognize that in graduate school, your job is to become a biochemist. And then your next choice will be either for the postdoc, or depending what industry you choose to go to Arzeta, you know, go to a company that's trying to do, let's say, protein engineering for making renewable materials or something like that. Or studying extremophiles, or, you know, there's a lot of chemical isolation from plants that's really productive. There's quite a few labs that are just stars at doing this. I'm trying to think of my colleague's name. He just started an institute at Northeastern University where they use Chinese medicine still as an inspiration to isolate compounds from nature. And these are very, you know, new compounds. We don't know what enzymes make them. And so aside from the usual argument for biodiversity that, oh, there are all these compounds that are good for our health, more than that, identifying the compound and working back to finding the enzymes is actually really important, because it gives us an idea of the diversity of enzymes that we can exploit to try to fix our climate problems. So that's something I've learned from giving this class, actually, is the value of learning about the enzymes. I'm just seeing that there's some stuff in the chat. Thank you for a great lecture. Oh, you're welcome. Peru's lab director, Alvin Kohler, is trying to understand the desiccation process in tardigrades. Okay, good. I didn't realize he was working on that. Look him up. I've been following this guy, Thomas Boothby. He's really amazing. He's a really nice guy. He's actually Swiss. Bizarrely, he works in Laramie, Wyoming, and he looks for all intents and purposes like a cowboy. But and he's one who taught me how to find tardigrades in the woods. I honestly, a surprise for me in this class was taking kids out to the forest. A lot of these kids who are, especially maybe at Yale, because they're really driven to go to the elite university and go to medical school or whatever, they have honestly, I tell them, get some of that pond scum and put it in a 50 ml tube. And they're like, you don't want me to touch that pond scum, do you? And I'm like, yes, I do. Go touch that pond scum. And they just not interacted with nature. And they're gobsmacked. They just love it. They're so amazed. And it makes them more interested to show them nature.
Yes, I think, yes. Just
Peter,
you can go ahead and ask questions.
Can you just, I asked before, but can you tell us again, what is the course or the general study that your students are coming from? Is that a biochemistry
Yeah, so in the US, you do it's called liberal arts. It doesn't mean that you're a humanist when you get out. You know, STEM is big time, like you can be a major in physics, chemistry, biochemistry. So I did biology with a concentration in neuroscience. So you're taking classes more broadly. You have to take English and a foreign language and all those things, but so you're trying to become a bit of a student of the world, but then you're also learning a discipline. And there's a real mentality in the US about what is the, you know, what are the major principles of the discipline. And so you are also supposed to work in a lab. So undergraduates, most of the time they have worked in the lab at least for a year. Oftentimes they publish undergrads. So these students are mostly biochemistry majors. I've also opened it up to environmental studies majors, which is kind of an interdisciplinary major at Yale. And they have been excellent students because they have the motivation from the living world, right? Like they know what a plant is and so on. And they know enough biochemistry to get into my class. I want to make sure that they're not overwhelmed because it's hard to read the papers if you don't have a biochemistry background. But they have been amazing. They really bring a new perspective to the class. So that's also very cool to have the biochemist, you know, biochemists are, you know, probably 80% of the students. And then they get to interact with these students who are, you know, going out in the field, doing a field experiment, collecting lizards, which respond to the temperature by changing the composition of their little gluey foot pads, which becomes really an interesting topic as well. So I just really feel like it's an enriching experience for people who are being trained in the classical discipline of biochemistry. It's, you know, we're not taking away from their training. In fact, we're teaching them how the planet even survives through nitrogen and carbon dioxide fixation. Oh my God.
Right. Thank you. Yes. Just as a side comment, I think this lecture is super inspiring. I would love to take it actually as a student. It's amazing.
The students, so many students say it's the best course they've ever taken. And I'm so happy about that. It's not about me. It's about the material. The material is so exciting. It's so exciting to figure out how biochemistry causes that, you know, the living world. It's just great.
No, it's amazing. I also think about the Austrian study system and how such a course could find its place and reaches a lot of students, and not only the ones that are specifically interested in saving the world, but actually those that haven't considered it at that point before getting into the course.
But you must have upper level electives, right?
Yeah, I think the tricky part is that the bachelor course is already so full that it's hard to squeeze something in. And for the masters, there are dedicated courses, but I think nothing yet in this kind, while it would be possible to put it in, yes. But
you want to get him before the masters because you want to convince them to keep going in biochemistry.
You're very right. Yeah.
So there's a hand up.
Hi.
Aaron. Hi.
This is Aaron from Max Peruf's labs.
Aaron.
I really liked this talk. I just wanted to ask something a little bit more personal, maybe. So we do a lot of cell culture and tissue culture. And, you know, it's not uncommon that at the end of the day, we feel like a ton of plastics, biosafety disposals at the end of our day.
I know, terrible.
That this like internally, like I don't want to say guilt, but like this, like, okay, I'm doing something useful as an experiment, but I'm also creating like so much plastic.
I know, it's horrible.
I'm sure you thought about this this dichotomy, and how do you resolve this, like this cognitive dissonance?
Well, this is something I want to work on this summer. One thing is we always had glass pipettes, you know, the sucker pipettes, as well as the, you know, 25 mil mouth pipetting. I love mouth pipetting. Anyway, glass. Anyway, very bad. We need to bring back those glass pipettes. I mean, it does take water to clean them. So I don't know what the carbon budget is surrounding that. So I think to some extent, we have to accept that the plastic plays an important role in our society, and we need to create these enzymes for breaking them down. You need to be able, the biochemical issue is the enzymes have to function at high temperature because you have to melt the plastic in order for the enzyme to get access to the plastic, right? So you want the enzyme to work at 60, 65 degrees, and it has to work at industrial scale. So it wants to work on the plastic in a day. So you start with hydrolases that might take a week to start to degrade the plastic. And then you engineer them to a hydrolase that'll work in a couple of hours at 65 degrees. It's so cool. There's such cool papers on this. It's like, touchdown. It's so cool. Anyway, so we need to have these tools to get, PET plastic's the worst one. And also to get the microplastics out of the place, the health effects of microplastics, the whale killing plastic. In any case, I know what you mean. It's guilt and sadness is what you're feeling. It's a feeling of loss, like when somebody dies, plastic kills whales. I mean, I think about whales dying and I feel a sense of loss. It's horrible. And so we need to learn what the carbon budget is of our labs, and we need to try to mitigate as much as possible. If it means going back to washing our pipettes and autoclaving them in canisters, I'd be really thrilled to do that. And but I don't know, like pipette tips, we go through so many tips and plates. Like, what about all those plates for the tissue culture cells? I totally agree with you. I don't know what the answer is. It's my summer project. Maybe I need to come back to you. I know there's a bunch of people trying to do this. In fact, there's the person who had the course. Who is the person? They were on the call before. Jerome has a class. He's probably fallen asleep by now. You have a class on lab sustainability.
Yeah, I'm still here. I'm just
tell us what
on
we
the
should
terrain.
do to reduce our carbon footprint in the lab and the plastic production.
Yeah, I mean, there's lots of initiatives already, but it's just scratching the surface, right? So we're not there yet.
So is there something you can do to, I don't know, disseminate the resources that are there? Somebody just, Nicola Winter just posted a link that I'm gonna, reuse of laboratory utensils reduces carbon dioxide equivalent footprint and running costs. Thank you. That's great. Okay, so Jerome is not responding. Okay. No, I think we have to do more. Totally. Also, what about how much we travel? I have a lot of guilt about traveling. I go to conferences, I get invited, and it's like, I'd really like to go to that conference. I'm going to Copenhagen today to a conference. I'm going to burn all that carbon dioxide. I had an idea that we could, scientists could have an agent, and they could like travel 1 month a year. They'd say, like, okay, I'm going to spend September in Europe. And then my agent could line up talks at all, possible universities and institutes that might want to hear a talk from me. And I might hit a couple of conferences along the way. So I could take a boat. Honestly, I'd take a boat. Potentially, that's another thing I want to look up. Is a boat better or worse than a plane? I'm not 100% sure. But I actually could write a grant on the 6 days it takes to take a boat over to Europe. And then I would travel to all these cities on the train. And then I would go back, and then that would be a much smaller carbon footprint than me going like this year. I'm going to be going to Europe in this month, September, October, back and forth over the Atlantic, burning up all that carbon dioxide. I feel terrible. But I also feel like there's a benefit to it, kind of as we were saying about using plasticware in the lab, obviously disseminating our research is also important. So I don't know what people think about that idea about having an agent and trying to concentrate our travel into like 1 or maybe 2 trips instead of this incessant several times, a month flying around to different places. Personally, I think that'd be great if I had an agent also. I wouldn't have to do all this emailing with all these different people and coordinating my own travel. The agent could figure it out. I just have to make one trip and then get a bunch of train tickets. It'd be great. I'm like, sign me up. I tried to convince my son to start a company doing that, but he so far hasn't bitten on that. Okay. Well, actually, I have to get ready to go on my flight to Copenhagen, so I think I better go.
Yes. So
Yeah.
Valerie asked me to kind of take over her role. So I thank you very much for having given this lecture to us today and to be open for these many questions and to
Here's what
engage
I'm
in
going
this
to do.
dialogue.
I'm going to share my, so what I had a hard time like figuring out what's the right way to package each lecture to make, so if somebody wanted to do a class similar to mine, like obviously, my recommendation as a teacher is you have to make the course yours. Doing a karaoke of someone else's lecture is not going to work. But just seeing an example. So I have all these reading lists for each of my lectures. I have a reading list that's the readings for the lecture, but also other papers of interest. And they can take like clues from that for their papers and so on. So I could like put the reading list in a Google folder, a Google Drive thing, and share that if that's interesting to anyone who wanted to think about, and for postdocs, it should be really interesting, right? Because they're going to get a job somewhere. If they get a job in a university, they're going to have to teach. And maybe it's exciting for them to continue in academia if they knew that they could potentially use some of this material in their teaching assignment. And so maybe that would be helpful to postdocs envisioning their future. What are they going to do?
It would be super helpful if you could share the background material for your lecture. I mean, I completely agree. Doing a karaoke would never work, but it's super helpful to have all this massive amount of papers and the knowledge that you put together. It's amazing. It's very inspiring. Yeah.
Okay. That will also be my summer project. We'll put this together in a digestible way for others to benefit from.
Super.
Okay.
That's great. Thank you very much.
You're welcome. Okay, I'm going to go pack my suitcase now.
Bye-bye, Karla.
Thank you very much.
Thanks for coming,
Bye.
everyone. I really enjoyed the discussion.
Bye-bye.
Bye-bye. Thank you very
Bye.
much.